are going to start the last part of this session about brain lesions and how we can learn from brain lesions how different parts of the brain are working. In the history of cognitive neuroscience, we have many famous cases of injuries in the frontal areas, in the temporal areas, in the parietal area, or in the occipital area that help us to understand how different parts of the brain are working. This really nice slide is provided by one of my colleagues, Dr. Rezapour. We are working on a kind of neuroscience textbook, and we are kind of discussing about these kind of cases. And we'll discuss about those cases together in the next chapters and next sessions that we'll have. But these cases are informing us in terms of how different parts of the brain are working. We will discuss about this specific case, Finas Gage, and how the damage in the prefrontal cortex could change the normal behavior. So we'll kind of use these cases to learn about how different parts of the brain are working when we have an injury in one part of the brain. In most of the cases, we have different patients, patient 1, patient 2, patient 3, that they have the same symptoms. For example, lack of control. So patient 1 has lack of control, patient 2 as well, and patient 3. But they have different lesions. Because we are not making the lesions are not in our control. People have different shapes of lesions, okay? But at the end of the day, we can get sure that, okay, they have overlaps, and probably if all 3 patients have lack of control, probably the area that is within the kind of the overlap of these 3 areas is the area that is responsible for lack of control. This is the way that we technically kind of use these cases to understand how different parts of the brain are responsible for different functions. But it is not always the case. Sometimes we have 3 patients, they all have the same symptom, let's say lack of control, but they have different areas of the brain that are affected. So they all have lack of control, but there are different areas. And that makes things a little bit complex. Why we have different areas? Why we have the same symptoms? Symptom is the same, but there are different areas. And that is something that we have in neuroimaging. Sometimes we have kind of a report about what are the brain areas that are involved in migraine. So there are reports that there are in lateral part, in the kind of temporal part, in the parietal part. So there are different reports, and sometimes you get just confused at why we have reports from different parts of the brain, and then we have the same symptoms. The reason behind that goes back to what we call the connections of these areas. How areas are connected together in the brain is going to be critically important, and we need to start to learn how different parts of the brain are connected to each other. I really like to kind of introduce a gentleman, Dr. Fox from Harvard University. He's doing a really nice job in terms of putting these pieces together. He has a really nice paper in New England Journal of Medicine, kind of describing these cases and how our understanding about brain networks would help us to have a better understanding about why these kind of variations that we have in the relationship between injuries and symptoms. Dr. Fox is right now the co-director of Branson Allen Brain Stimulation Center in Harvard. I took my kind of short fellowship on brain stimulation there in 2009, something like 11 years ago. But they are doing really nice. They are one of the leading centers that use these kind of ideas to help patients in the process of treatment. So if we kind of consider the connection between these 3 areas, in reality we'll realize that they are connected in a network that we called executive control network, what we called ECN. They are all nodes of the same network. Whether we measure that with DTI or anatomical connectivity, they are connected to each other, or whether we use functional connectivity with fMRI. So this is fMRI, This one is DTI. With all these measures, we can see that these areas are connected. And they all play a role in the executive control network. So that is the reason that all these 3 patients could have the same symptoms. And these areas that are in the executive control network, I mean, this is another atlas, but you can see those basically orange areas. These areas are a part of the executive control network. When they are connected to each other, they just oscillate in the same rate. So this is called what we call, they are something like phase lock. They are locked together and they oscillate together. When they start to oscillate together, they get connected and they start to transfer information together. Okay. One of the idea is, okay, can we synchronize these areas when they are not synchronized to get them connected? And that is what we are doing right now in my lab. So we put electrodes over their head, and as you can see here, this is one of my kind of assistants, Ashkan. He's a kind of electrical engineer, and he's a really genius guy. He's helping us to put all those electrodes over the head, and then we can basically try to stimulate these areas in a way that they oscillate together, and when they oscillate together, they can get connected to each other. We are running a trial right now among opioid users to help them to improve the executive control function by just connecting these areas together that might not be connected well together. And so it is not just about stimulating one area, it's just about stimulating a network. And this is not only stimulating a network, but also synchronizing different nodes of the network in a way that they can get connected. So that is what we are hopeful to use. And to be able to find the best areas that are going to be connected, this is exactly what we need. We need to have those connectograms that we discussed last session about how these areas are connected together. So we need to have a knowledge about how these areas are connected and how these connections even are impaired in different injuries. We have a case that is called Finas Gage, who was working in kind of the railroad constructions, and he was using an iron kind of bar, putting dynamite in a hole, and the dynamite exploded, and the iron bar went from the kind of the left eye and the kind of the lower the left eye, just going into the kind of into the eye, and just going to the kind of prefrontal cortical areas and damaging all parts of the prefrontal cortex. And how that damage changed his behavior and the things that was happening to him. He's dead right now, but if we have the case like this, then we can even measure the connections that we still have in the brain and the connections that are impaired with these connectograms. And then when we have these connectograms, we might even start to think about, okay, if I want to do a stimulation, if I want to find the areas in the brain that are going to be stimulated, how these connectograms could inform me in terms of helping the patient that already has some disconnections in these networks. Okay? To be able to understand these connections, we need to know about these networks. And that is going to be something I'm going to cover for the next session. For example, we need to know how these kind of white matter tracks are going into the brain. So it is not just about, as we discussed in the previous session, it's about just the gray matter areas. It's also about the white matter areas, how left and right brain are connected together with corpus callosum, or some other top-down areas in the brain. This one is corona radiata. So it is another white matter tract that goes inside the brain, and it goes to a really small bottleneck here. It basically brings information from the cortex down to the spine. And when we do neurosurgery, if you just cut a small piece of that bundle here, that would make a significant kind of disaster. Just cuts down several of those fibers so it could at least make something like hemiplegia or different sort of parastasis. So it would just can make people paralyzed in one part of the brain. So there are some kind of negative consequences because you have lots of fibers that could go into a really small place, which is called internal capsule. So this part is called the internal capsule of this gradation in the brain. And then the last piece of the information I'm going to give you today. I've already bombarded you today with huge amount of information. I know that we have discussed about many things today, but I mean, we are going to remain strong and kind of learn about those things over time. The last piece is about a really interesting kind of pathway, white matter pathway. It's called arcuate fasciculus. Why it's called arcuate? Because it's something like an arc. It is something like an arc, okay, because it's called arcuate fasciculus. How it is working? It is basically connecting the Broca area to the Wernicke area. Broca to the Wernicke area. This is where we understand language. This is where we speak with. And this connection is critical for us to be able to talk. This is the last paper I'm going to show you. This is a paper that is right now in bioRxiv. By having that in bioRxiv, it means it hasn't published yet. This is a preprint. It is not an article yet. It has been posted just a few months ago. It is going to be published. It's from a group of people that are doing a really nice job in Pittsburgh University. So they can kind of detect its arcuate fasciculus, and they have developed a really interesting pipeline to develop, kind of find this pathway in the brain. And it's not just finding the pathway. They can measure the diameter, they can measure the length, the radius, the span, and they can clearly show that how these different people have different measures for these items in their brain. One of the postdocs in our center, because I saw that on Wednesday, we had a meeting on Tuesday. Haisha, one of the postdocs in our center, was testing that in 364 subjects in one database and 264 subjects in another database. For the arcuate fasciculus, the diameter, okay, in the right and left lobe, basically left brain and right brain. And you can see here that with this kind of really strong effect, this is the Cohen D of 1.6, it's a really strong effect. In the left side, in both groups, in the left side, the diameter is higher. Why? Because why do we have such a kind of larger diameter on the left side? At least most of the patients.
Dominant hemisphere. Because of dominant hemisphere is the left
because
side.
that is
Exactly, because these people are using this side for the language. And that is, but it's quite interesting that there are people who are here. There are people who are here. So people are not exactly the same. So people are different in terms of, even within the group, you can see that there is a variation in terms of the shape and the size of this one. And it might be also interesting that when I show you different brains of different animals, okay, this is one of the things I'm going to show you in the next sessions as well. This is the elephant. This is the elephant. This is the elephant. Elephant. This is dolphin. So you can see that how, and this is us, so human being, okay? So if I just show, this is a chimpanzee brain. It's amazing that in chimpanzee brain, we have both Broca area and Wernicke area, okay? These two areas are here in both chimpanzee and human being. The only difference is that these two areas are connected in human, but they are not well connected in chimpanzee. And chimpanzee doesn't have language. And it seems that there might be just minor genetic items that would contribute in development of this pathway. And then the $1 million question is, okay, if we do those genetic modifications in the chimpanzee's genes, can we make these two parts in chimpanzee brain connected? And if we can make them connected, can chimpanzee talk or not? So that is the beauty of cognitive neuroscience. And just the last slide, just showing you kind of the few kind of subjects to show you that how much they have different shapes in their arcuate fasciculus. I'm not sure if you have this type of arcuate fasciculus or this type. I'm sure this person is a really, so these areas are really highly connected. Or so you can see that people are really different in terms of how they have arcuate fasciculus. And the beauty of cognitive neuroscience is we can assess people, how they are talking, how they are interacting, and then measure how these kind of connections inside the brain are there, and then how they are related to each other. That is going to be the last slide of my talk today. I hope that I have not made you really exhausted today, but this is a really exciting area of seeing how, and what I really kind of highly encourage you when you kind of get a PhD or postdoc position in these areas, just one of the first things that you do, just take a kind of a brain image from yourself, and when you do those kind of measures, just do it for yourself to see, okay, which one is where is my kind of the arcuate fasciculus? What are the features that my arcuate fasciculus have? That's the interesting thing about the brain. We usually see, let's say, okay, I have strong muscles in the arm, I have kind of longer bones here. We can see what is happening in the body. We have a clear idea about the size and shape of different organs that we have outside the brain. But we do not see those parts in the brain. And then the cognitive neuroscience would give you this opportunity to see what is happening in the brain and how they are important in terms of, okay, how we behave and how we might be able to help patients who are suffering from some disconnections inside their brain. Okay, good. Okay, so I'm really happy that we have this class with right now people from 24 different countries. So it is amazing that we have people from different countries here. And I really appreciate that we have a really strong cultural, religious, ethnical background here, diversity here. And that diversity is something I really appreciate. And I really like those who are from different countries to talk in the next sessions about their cultural experiences, about what are the things that are significant for them from the cultural perspective. For example, I know that we have people from different countries that they have kind of Shia Muslims there, and today was a really significant day for them. I received kind of an email from one of the people in the class. I'm not sure if Leila is here, Leila Nurbala. If you are here, if you can turn on your video, she's going to give you an idea about why this is significant to Shia Muslims this day. And I'm going to ask others who are in the class to do the same for other kind of religious background. If we have from Sunni Muslims here, we have Hindus here in the class, we have people from Christianity, different Christianity schools, different branches of Christianity. We have people from the Jewish background. We have people from even different kind of African kind of tribes or African countries. So whatever people have in terms of their religious background, their cultural background, their ethnical background, and they want to share those things, just kind of send me an email, and I will kind of give probably 1-2 minutes to those people who are interested to share their specifically kind of to that specific week, what is happening at that specific week that is significant for their nation or for their kind of their group. That is going to be something that we are going to do for the next session as well. Great. Thank you very much, everyone, for listening and being a part of the class, and I hope that you enjoyed that. Have a good day.